So what if the cure for a chronic disease, doesn't actually reset your body to normal?
Right, which is what we all assume happens.
Exactly. I mean, what if your immune system remembers the trauma of a viral war so deeply that even after modern medicine completely wipes out the virus, your cells are just left with this permanent molecular scar?
It's a wild thought. It really challenges everything we usually assume about, recovering from an illness.
Yeah.
We tend to think that once the pathogen is gone, the body just naturally goes back to baseline. But the microscopic reality is far more complicated and, honestly, much more fascinating.
Well, welcome to the deep dive. Today, our mission is decoding a really dense, just groundbreaking new paper from the journal Immunity, published by Reinscheid and colleagues.
A fantastic paper.
It really is. We are looking at exactly how the immune system forms memories of chronic infections and the specific imprint those infections leave behind on you. And to do this, we are focusing on the generals of your immune system army, your CD4 plus T cells. Now, I know the CD8 plus killer T cells usually get all the glory in these exhaustion studies, right?
They definitely do. They're the frontline infantry.
Right, but the CD4 plus cells, they're the crucial orchestrators. They direct the whole war effort.
Exactly. Without the CD4 plus generals providing, strategic instructions, the CD8 plus soldiers essentially lose their coordination. They need that command structure.
Right. So what's our model for studying this?
Well, the perfect human model to study these generals is hepatitis C virus, or HCV.
Why HCV specifically?
Because it naturally provides us with 4 distinct war scenarios in humans, which is incredibly rare to find in just one disease.
Oh, interesting. What are the 4?
So we can look at acute infection, then spontaneously cleared infection, which is where the body wins the war naturally.
Right, without drugs.
It's exactly. Third is chronic infection, where the war just drags on for years. And fourth is therapeutically cured infection, where the war is ended abruptly by modern drugs called direct-acting antivirals, or DAAs.
Okay, I want to set an expectation for you listening right now. We're going to examine the mechanisms of this paper in extreme detail.
We're going deep.
We are leaving no stone unturned. Because to truly understand the imprint chronic disease leaves on your body, you have to understand the microscopic machinery at play.
You really do.
So let's start with the sheer logistical hurdle of this research. How do scientists even study these specific CD4 plus T cell generals? Because, in a chronic HCV infection, the specific CD4 plus cells tailored to fight that exact virus are incredibly rare.
They're vanishingly rare, like needle in a haystack rare. In a chronic infection, finding them is a massive challenge. So to solve this, the researchers relied on a highly advanced technique called PMHCII tetramer technology.
Okay, PMHC what? Let's break that down.
Right, sorry. It stands for peptide major histocompatibility complex. Usually, T cells recognize viral fragments, which are called peptides.
Right.
But they only recognize them when they are presented on the surface of other cells by this special protein complex, the MHC class II.
Okay, so it's like a display case.
Yes. The perfect analogy.
Got it.
So the researchers artificially engineered these display cases in the lab. They loaded them with specific pieces of the hepatitis C virus, and then they attached them to a fluorescent magnetic marker.
Wow. So it's essentially molecular fishing.
Exactly.
You're dropping this highly specific magnetic bait into the bloodstream, designed so that only the exact CD4 cells biologically tailored to recognize hepatitis C will bite.
That is a perfect way to visualize it.
Yeah.
Only these specific HCV targeting cells bind to the engineered complex. And then the researchers literally use a magnet to physically pull those specific cells out of the blood sample.
That is amazing. Okay, so once they caught these rare generals, what did they do with them?
They didn't just count them. They used 2 really powerful tools to read their diaries, essentially. Flow cytometry and single-cell RNA sequencing, or cRNA-seq.
Okay, let's take those one by one. Flow cytometry first.
Sure. So flow cytometry uses lasers to bounce off proteins on the surface of the cell. Proteins like CXCR3 and CXCR5.
And what do those proteins tell us?
It tells us what uniform the cell is currently wearing, which indicates what it's equipped to do on the outside.
Okay, so if flow cytometry is looking at the outside uniform, single-cell RNA sequencing is looking inside the command center, right?
Precisely. cRNA-seq goes inside to sequence the messenger RNA.
The mRNA.
Right. DNA is the master blueprint, but mRNA represents the active instructions the DNA is currently sending out to the cell's protein factories. By reading the mRNA, we can see exactly what the cell is planning to do.
It's revealing its polarization or its fundamental identity.
Exactly. And here's where the paper reveals its first massive surprise.
Oh, I love a good science surprise. Lay it on me.
So, classic immunology textbooks tell us that CD4 plus T cells differentiate into mutually exclusive lineages based on the threat.
Right, like Th1, Th2.
Right. Th1 cells fight viruses, Th2 fight parasites, and Th17 fight fungi.
So naturally, you would expect the cells fighting the hepatitis C virus to be polarized as Th1 cells because it's a virus.
You would entirely expect that. But in chronic and cured HCV, the sequencing data showed these cells overwhelmingly had a TFH-biased profile.
Wait, TFA, that stands for follicular helper T cells, right?
Yes, it does.
But that flips the textbook upside down. If they are acting like follicular helper T cells, doesn't that mean they're abandoning their primary antiviral programming?
It seems like it, yes.
Because TFH cells usually hang out in the lymph nodes to help B cells make antibodies, don't they? Why would virus-fighting generals shift to that profile?
It suggests a really profound adaptation. During a prolonged, exhaustive, chronic viral siege, the immune system fundamentally rewires its generals into this unique state that blends features of both viral-fighting Th1 cells and B-cell-helping TFA cells.
So it's a hybrid.
Yeah, it's a survival and maintenance tactic. It keeps the immune response going without the cells completely burning out. And we simply couldn't see this nuanced blend before we had the resolution of single-cell sequencing.
That is wild. Okay, let me pause and make sure I'm fully wrapping my head around this setup before we look at the actual chronic phase. Let's do a quick recap for you listening.
Sounds good.
Okay, number 1. CD4 cells orchestrate the immune response. Number 2, researchers use engineered molecular fishing lures, the tetramers, to isolate these incredibly rare cells. And number 3, reading their internal mRNA reveals that they adopt an unexpected TFH-biased profile just to survive a chronic infection.
Nailed it.
So I want you listening to think about this question. If you were designing an immune system, why would you shift your generals into a hybrid profile during a never-ending war instead of just keeping them purely focused on direct combat?
It's a great question. And that underlying logic of survival perfectly sets up what the single-cell RNA sequencing revealed next.
Okay, where do we go next?
Well, when the researchers took all that sequencing data, they ran it through a computational algorithm called Leiden clustering.
Leiden cluster. Okay, think of that like a high-dimensional sorting hat, right?
Yes, exactly like a sorting hat. It mathematically groups individual cells based on the similarities of thousands of different genes they're actively expressing.
Right.
And what it showed was that during a chronic infection, these CD4 plus T cells aren't just one big, homogeneous, exhausted group of soldiers.
Okay, if they aren't one uniform group, what did the algorithm sort them into?
They divide into two very distinct, highly specialized camps.
Tell me about Camp 1.
Camp 1 is what we call stem-like cells. The transcriptomic data showed these cells heavily express a specific gene called BCL2.
BCL2, and what does that do?
It's a classic survival gene. Its primary function is to actively prevent the cell from undergoing apoptosis.
Which is programmed cell death.
Exactly. It keeps them alive. These cells also heavily express a transcription factor called TCF1, which acts as a master regulator for maintaining a state of stemness and deep quiescence.
Quiescence. So they are resting. They are progenitor cells.
Precisely. They are sitting out the direct fight.
So if Camp 1 is a resting stem-like bunker, logic tells me Camp 2 has to be the actual infantry taking the hits. What do those frontline cells look like under the microscope?
You hit the nail on the head. Camp 2 consists of the effector cells, and their genetic readout looks entirely different from the stem cells.
What are they expressing?
They express a gene called TBET. This is a master transcription factor that drives cells to actively fight and produce inflammatory cytokines.
Okay.
They also express CD38, which is a marker of intense cellular activation. Yeah. And they express Ki67.
Ki67, I've heard of that in cancer diagnostics, actually.
Yes, because Ki67 is a protein that is only present in the nucleus when a cell is actively multiplying and dividing.
Oh,
so Camp 2 represents the active frontline combat cells. They're constantly dividing, fighting, and ultimately showing all the signs of being exhausted by the constant viral assault.
All right, to help you listening lock this in, let's use a quick memory trick. Let's use the letters. Think of the gene BCL2 as the backline base.
I like that.
Right, these are your resting, stem-like progenitor cells safely away from the immediate fighting. And think of the gene TBET as the trench battlers. These are your active, exhausted effector cells fighting the virus on the front lines. So BCL2 is the backline base, and TBET is the trench battlers.
That imagery perfectly captures their roles.
Mhm.
But what's really vital here is understanding the structural relationship between these two camps.
What do you mean, like, if they talk to each other?
Right. Are they completely independent lineages, or do they interact? To answer this, the researchers used a computational technique called pseudo-time trajectory modeling.
Pseudo-time trajectory. That sounds like time travel?
It essentially is, for cells. This algorithm calculates the transitional states of cells, basically drawing a map to show how one state gradually evolves into another over time.
So it tracks their lineage.
Exactly. And they combine this mathematical modeling with something called TCR clonotype tracking.
TCR being the T cell receptor.
Right, the specific protein complex the cell uses to recognize the virus.
Yeah.
Now, because the genetic sequence for this receptor is unique to a specific lineage of cells, it acts like a permanent barcode.
Okay, I'm following.
By tracking these barcodes across the two camps, they definitively prove that the stem-like cells, your backline base, are actively giving birth to the effector cells, the trench battlers.
Right. Okay, let me trace the logic here. We have an underground bunker command safely away from the front line, and it is constantly deploying new soldiers because the frontline troops just keep getting exhausted and dying off in this chronic viral siege.
That is exactly what's happening.
But wait, I have a question. If this is an adaptation to a chronic infection, does this bunker only get built after the war drags on for months and the body finally realizes it's losing?
It's a highly logical assumption. But the data showed something incredible, and this raises a crucial point about how proactive our immune system actually is.
Oh, really?
Yeah, the researchers found evidence of preemptive formation.
Preemptive.
By analyzing blood samples from patients in the very acute early stages of infection, before it ever became chronic, they discovered that this backline base bunker is built almost immediately.
Wow. So the body prepares for the worst-case scenario of a forever war from day one.
Yes, from the very beginning.
That is just profound foresight by the immune system. Okay, let's synthesize this segment for the listener.
Let's do it.
Number 1, chronic CD4 cells split into two distinct camps. Stem-like cells expressing the survival gene BCL2 and effector cells expressing the combat gene TBET. Number 2, we know the stem cells act as a continuous factory, giving birth to the effectors. And number 3, we just learned the structural split happens preemptively during the initial acute phase.
Perfect summary.
So imagine your own immune system right now. What kind of viral bunkers do you think you're carrying around from past colds, flus, or other infections that your body thought might turn into a forever war?
It's slightly unnerving to think about, but incredibly efficient. And it leads us perfectly into the next major question.
Which is?
What happens to this carefully balanced ecosystem of the backline base and the trench battlers when modern medicine intervenes and artificially ends the war?
Yes, let's look at the therapeutically cured scenario. We mentioned direct-acting antivirals earlier, or DAAs.
Yes, for hepatitis C, these are frankly miracle drugs.
They really are. They don't just help the immune system, right? They directly interfere with the virus's internal replication machinery.
Exactly. They completely shut down the virus's ability to multiply. Within weeks, the virus is wiped out. The war is just over.
So what happens to our two-part army?
The researchers tracked patients longitudinally. They took blood samples for up to 8 years after these patients were completely cured by these DAA drugs.
8 years? That's a huge data set.
It is. And what they observed was dramatic. Without the constant presence of the virus driving the war, the effector cells, the TBET expressing trench battlers, they completely vanished.
So they just die off.
Yeah. Without a target to fight, the front lines were abandoned and those cells died off. But the stem-like cells, the BCL2 positive backline base, they persisted in the bloodstream for years. They basically transitioned into a state of long-term memory.
Okay, so the troops go home, but the bunker stays open.
Exactly.
And there is a specific case study in this paper that I found absolutely fascinating regarding just how functional this bunker remains. Walk us through the relapse patient.
Oh, this case study is essentially the smoking gun for their entire theory about the bunker's purpose.
Set it up for us.
Okay, so they had a patient who went through DAA treatment and achieved a full cure at the 12-week mark.
Awesome.
And exactly as expected, their active effector cells completely vanished from their blood, leaving only the resting stem-like cells. But 8 weeks later, this patient experienced a relapse. The virus managed to come back.
Oh, no. And how quickly did the immune system realize the war was back on?
The response was instantaneous. Those dormant, resting stem-like cells mounted a massive, aggressive recall response.
They woke up.
They woke up and they rapidly ramped up production of brand new effector cells. Suddenly, the patient's blood was flooded with cells expressing Ki-67 for rapid proliferation and CD38 for intense activation. The immune system went right back to war.
Wow.
And then, when the patient was given a second round of DAA treatment and cured for good, those new effector cells died off again, and the stem-like cells went right back to being dormant.
I mean, deactivating a military base is just the perfect analogy here. You send all the frontline troops home, but you keep the bunker fully operational. Lights on, systems running, just in case the enemy paratroops back in.
It's incredible.
And this specific relapse case study proves the bunker still has a direct, functional line to the weapons factory. The troops can be deployed overnight.
Exactly. It proves that the stem-like cells aren't just leftover, inactive debris. They are highly functional, dedicated memory units perfectly calibrated to respond if the chronic threat ever returns.
Okay, let's gather our thoughts on this medical intervention phase. Recap time.
Go for it.
Number 1. DAA cures eliminate the active effector cells, but allow the stem-like cells to persist for years as long-term memory. Number 2. The relapse case study confirms these stem cells remain highly functional, ready to deploy new troops at a moment's notice. So think about the sheer metabolic efficiency of this. Question for you listening. Why do you think the body explicitly chooses to keep the stem-like cells rather than keeping the fully armed effector cells on patrol after the threat is gone?
Well, it really comes down to biological cost. Keeping effector cells armed and ready requires an immense amount of metabolic energy.
It's exhausting.
It is. Plus, highly active immune cells secrete inflammatory chemicals that can cause collateral damage to your own tissues over time.
Oh, right.
So keeping the quiescent resting stem cells is biologically cheaper and much safer for the host body. But this actually brings us to the ultimate question of the paper.
Yes, the climax. We need to compare this medically induced memory to natural memory.
Right.
Okay, so we know the bunker stays operational after DAA drugs clear the virus. But that makes me wonder if the body clears the virus completely on its own in the acute phase, without drugs, does it keep that same bunker? Is an immune system that was medically rescued identical to one that won the war naturally?
Biologically, no. They are profoundly different. Yes. And this is where the paper's findings become genuinely revolutionary. The researchers discovered that the CD4 T cells from patients who were cured by DAAs carry what they call a chronic imprint, or a molecular scar.
A molecular scar.
Even years after the virus is totally gone, these medically rescued cells are fundamentally altered at a structural level compared to cells from someone who cleared the virus naturally.
What does a molecular scar actually look like in the data? I imagine it's not a physical mark, but a change in how the cell behaves.
Exactly. It looks like a hardwired genetic preference for resting over reacting. The DAA-cured cells heavily express genes associated with exhaustion and impairment.
For example?
For instance, they show a strong LAG3-mediated impairment signature.
LAG3. What is that?
LAG3 is an inhibitory receptor.
Yeah.
Think of it as a physical brake pedal on the surface of the T cell. When it binds to specific proteins, it blocks activating signals from getting through to the nucleus.
Because the cell is actively suppressing its own ability to fight.
Yes, the brakes are on. And they also show high usage of something called the partnerless NF8 pathway.
Partnerless NF8. Okay, explain that one.
Sure. Normally, the transcription factor NF8 TARC partners with another factor called AP1 to fully activate a T cell and trigger a robust immune response.
They work as a team.
Right. But when NF8 acts alone, without its AP1 partner, it drives a genetic program of exhaustion and unresponsiveness.
Oh, wow.
Because of these functional scars, when researchers extracted these cured cells and stimulated them in the lab, they produced significantly fewer protective cytokines compared to cells from patients who cleared the virus naturally.
So they are sluggish, they just don't want to fight.
Exactly.
And there was a fascinating nuance here regarding a specific gene called TOX, right? How does that play into the scarring?
The TOX gene is critical to understanding the depth of this scar.
Yeah.
TOX is a master epigenetic regulator.
Epigenetic meaning?
Meaning it literally changes how the DNA strand is folded and packaged, fundamentally altering which genes the cell can easily access.
It's changing the hardware.
Yes. Now, the surprising part initially was that TOX expression was high in both the DAA-cured cells and the naturally cleared cells.
Oh, so on the surface, they looked identical.
Exactly. But the researchers didn't stop there. They used an incredibly sophisticated computational algorithm called ScaleMiner.
Right, StingMiner. And that performs network inference, right? It looks for the hidden wiring.
Precisely. ScaleMiner doesn't just look at whether a gene is turned on or off. It maps out the regulon. meaning it identifies all the downstream target genes that a master switch like TOX is actively controlling.
It maps the whole circuit board.
Yes. And the wiring between the cured cells and the naturally cleared cells was completely different.
How so?
Well, in the medically cured cells, the TOX regulon was wired to genes like TCF7, forcing the cell's circuitry into deep quiescence and survival mode.
Okay, and in the natural ones?
In the naturally cleared cells, the TOX regulon was wired to genes like STAT4, which is a pathway that primes the cell for rapid, robust effector readiness.
So, let me make sure I'm getting this. If you win a war quickly and naturally, your generals are victorious champions. They are alert, their circuitry is primed for readiness, and they are eager for the next fight.
Right.
But if you were trapped in a grueling, multi-year siege and had to be rescued by a medical airstrike, the DAAs, your generals, survive, but they essentially have physiological PTSD.
That is a very accurate way to describe it.
Their fundamental epigenetic wiring has been changed. They carry molecular scars that structurally bias them toward resting and just surviving rather than aggressively fighting.
That is precisely what the spying Einer data proves. The sheer trauma of the prolonged chronic infection leaves a permanent mark on the internal architecture of the immune memory, completely changing how those cells function years down the line.
Wow. Let's synthesize this final crucial segment. Let's get our bullet points. Number one, immune memory after a medical cure is fundamentally biologically different from memory after natural clearance. Number two, these medically cured cells carry an epigenetic and transcriptional scar driven by the brake pedal LAG3, partnerless NF8T, and a rewired TOX gene network. And number three, practically, this molecular scar forces the cells into a state of deep quiescence rather than robust readiness.
Perfect.
So here's my question for you listening. If a medical cure leaves a permanent scar on our immune memory, how should that alter the way we approach treating chronic diseases? Does it mean early intervention is more vital than we ever realized?
That is exactly the takeaway. You want to intervene before the scar is formed. And if we zoom out to the bigger picture, the implications of this immunity paper are staggering.
Because it's not just about hepatitis C.
Not at all. The mechanisms we just walked through, this backline base of stem-like CD4 plus T cells, the exhaustion markers, the deep epigenetic scarring, these findings map incredibly closely to what oncologists observe in human tumors and what virologists see in other chronic infections like HIV.
Because a solid tumor is essentially a chronic siege, right? It's a never-ending war from the immune system's perspective.
Precisely. In cancer, the CD8 plus killer T cells constantly attack the tumor, they get exhausted, and ultimately fail to clear it. But if we can figure out how to pharmacologically manipulate these CD4 plus stem-like progenitor cells, the bunker command, we might be able to engineer a continuous unexhausted supply of fresh, potent effector cells.
That would change everything.
It really would. Understanding this specific CD4 plus structural split is likely the foundational key to designing the next generation of cancer immunotherapies or advanced therapeutic vaccines.
You know, we spend so much time and billions of dollars trying to develop drugs to just eradicate viruses or kill tumor cells. But if our immune cells carry the epigenetic and transcriptional scars of chronic infections, even after a complete medical cure, are we ever truly back to baseline?
Probably not.
Right. And could we one day design therapies that don't just kill the invading pathogen, but actually go into the nucleus of our own cells and erase the trauma from our T cells' DNA?
Being able to clear the molecular scar and reset the immune system to true baseline, that's the next great frontier of immunology.
It's an incredible thing to think about. Thank you for joining us on this deep dive. Keep questioning the incredible, sometimes murky machinery inside your own body. We'll see you next time.